Oh, you must be Victor. Here, have a seat over there. The doctor will be with you shortly.
Hi Victor, I'm Dr. Heisenberg.
Nice to meet you.
I've got your charts right here and your eyes are looking fine.
Oh wow, that's great.
Yeah, so you're free to go till next year.
Oh wow, thank you. But can I ask you something really quickly?
Yeah.
I've been working on this class project about how the eye works, and I'm having some trouble working on it. So I was wondering if you could give me some pointers or some hints on how to do it.
Yeah, I could explain those things to you, but it's a really long story and it'll probably take 5 minutes.
I love long stories. So.
Alright then.
Within our eyes, there are tiny cells called photoreceptors that absorb at specific regions of light and allow us to see. There are two types of photoreceptor cells, rod cells and cone cells. Rod cells are responsible for our night vision and work best under low light levels. There are three types of cone cells within our eyes that can cause color vision. The S cone is responsible for blue vision as it absorbs light photons in the blue region. The M cone absorbs light best at green wavelengths. And the L cone absorbs light best at red wavelengths. Under light conditions, these cells become active, and they respond by absorbing light photons. After a cell gets activated, a process known as phototransduction occurs and allows us to generate a visual image. Each photoreceptor cell contains a molecule called the 11-cis retinol that is responsible for this process. When a photon strikes the 11-cis retinol molecule, the molecule becomes photoisomerized and transforms into the all-trans retinol molecule. Here we have a rhodopsin protein. When it absorbs a light photon, the 11-cis retinol turns into trans retinol. This causes the protein to change shape. This leads to the phosphorylation of the G protein. The G protein then binds onto the enzyme phosphodiesterase. This complex then catalyzes cyclic GMP and cleaves them into GMP molecules. The reduction in cGMP concentration causes the ion channel pump to release the cGMP molecule, thus closing the channel. This causes the cell to become hyperpolarized, which generates a signal to the brain to produce a visual image.
Oh, doctor, how does the isomerization process actually work?
Well,
what happens is one of the pi electrons gets excited by the absorption of a photon. This weakens the bond and allows the bond to rotate from a cis conformation to a trans conformation. After the bond rotates, the pi electron gets relieved and drops back down in the ground state, thus stabilizing the trans conformation.
and that's how we're able to see.
But doctor, does retinol molecule ever change back?
Yes, it does.
What happens first is an enzyme known as RDH reduces the all-trans retinol into an alcohol. The retinol molecule then gets transported out of the photoreceptor cell and into the retinal pigment epithelium, where another enzyme called LRAT catalyzes the retinol into an ester molecule. From here, the molecule, with the help of an enzyme called isomerohydrolase, isomerizes and hydrolyzes into 11-cis retinol. Finally, the 11-cis retinol molecule is oxidized into 11-cis retinal and is then transported back into the photoreceptor cell, where it attaches back onto the opsin protein.
And that about wraps it up.
Oh wow, thanks for your help, doctor. It was really helpful to know that, and that was a really cool story.
No problem, Victor. So if you have any more questions, feel free to ask, but otherwise I'll see you next year.
Yeah, thank you for your help today.
No problem, I'll walk you out.
Yeah, thanks.